I'm all right. I'm going to show some slides for about 10, 15 minutes, background on orthology and ortho MCL, and then take you through a little tour of ortho MCL. Um, I'm going to go with the desktop share because that's been working for me and make my ortho slides full screen. Um, One thing that I want to say is that when I'm doing the tour and some of my overview is going to be based on the new version of Ortho MCL, which right now is available in beta. We have this little banner on top. When you're on the genomic sites, all PlasmoDB, et cetera, and you link through from a gene to its ortho group, you will be going to Ortho MCL version 6, which is our production version. And you have to click on this banner, Ortho MC. Yeah, well, click on the banner and take yourself over to Ortho 7 if you want to see the newer features that I'm going to talk about. Okay, just a little background. We've seen the synteny plots before, and just in general, what are we talking about when we talk about orthologs? And the simplest definition I have come to is that orthologs are the same protein in a different species. So when you're looking, like in a synteny plot, at closely related species that might be in the same genus or the same family, they're expected to have the same set of proteins with relatively small changes, both in the protein set and in the amino acid sequences of the individual proteins. However, we can find orthologs, the same protein with the same function, in more distantly related species, particularly for highly conserved processes, glycolysis, DNA replication, etc. Um, why do we have ortho-MCL in ViewPath? Why is it useful and important? Orthologs have a conserve function that often provide the best information when you're trying to annotate genes that don't otherwise have annotation. This might be the newly predicted genes in a newly sequenced organism, or it might be that an experiment has been done in one organism that does not have great annotation and you want to benefit from annotation in another organism. So, or you've done an experiment in a model system and you want to translate those results to another organism where perhaps you need to develop a drug. So in both cases, we are looking to find the equivalent genes in another organism based on a list of genes in a particular organism where we've done some type of computational and genomics experiment. In ViewPathDB, we have this as an orthology transform function as a search step. So in a search strategy, you have a list of genes. You can transform that list of genes into related records in another organism. Oh, um, this idea of same gene in another species is complicated in eukaryotes due to gene duplication within genomes. So you can have multiple copies of a gene. There can be recombination so that parts of one gene can combine with parts of another. Those could be genes that are very similar, or they could be genes that are quite different, so that you could have, say, an additional functional domain added on to a protein in some species. Then there's speciation, where we're creating new genomes that can then evolve separately and develop new sequences. And gene loss can also occur. Either the only copy of a gene can be lost from a particular lineage, or some copies of a multi-gene family can be lost so that we no longer have a perfect one-to-one correspondence. And a classic example of this is the globin gene family, where we have many gene duplications that have happened at different times in evolution. And so we have alpha globins, beta globins, myoglobins. And when you look at the complement of genes in a variety of different organisms and you try and match them up, what is exactly orthologous to what, it becomes quite a complicated process. In ortho-MCL, we include both the orthologs, the identical genes that match up in a one-to-one way, and also paralogs, which are genes that have been duplicated within the same genome and then passed down as gene families due to subsequent speciation. So to deal with both of these types of gene copies, we create groups, which we call orthogroups, which contain both the orthologs and paralogs across many species. We use a sequence similarity tool between the amino acid sequences of the proteins using a tool such as BLAST. Actually, we do use BLAST. Other orthology algorithms also rely on similar sequence similarity. However, when dealing with a lot of data, all the proteins from a large number of species, we call them proteomes in each species, all the genes, genome, proteome, the computation grows exponentially large because you're comparing every single protein to every other protein. ViewPathDB has over 700 species and strains, and OrthoMCL has over 8 million proteins. To simplify this computation, OrthoMCL has developed an algorithm with a two-step process. We've chosen 150 species that are taxonomically well distributed across the tree of life, and we've designated them as core species. And we've taken those 150 proteomes and done this all versus all similarity analysis and put them into groups that we call core groups. And then we add in all the other proteins from all the other species into those core groups. I should mention that in the new version of OrthoMCL, the clustering step is done by a software called OrthoFinder, which is, we believe, an improvement from the OrthoMCL algorithm that we've used in previous versions. So this set of 150 diverse species we call the core. We add in all the other ones, and then the leftovers, those that don't match any protein in any of the core clusters, are themselves clustered into what we call residual clusters. So we have a large number of these orthogroups, and we have a set of tools that allows you to find orthogroups that meet your interests, a particular set of criteria that might be interesting to you. Obviously, you could search by a gene. What cluster does my gene fall into? If you know a gene, you can search by a group ID if you know an OrthoMCL group ID, both in the current version and group IDs from past OrthoMCL versions. You could search by a text term, something in the description of a protein, PFAM functional domains, and we also have something called a phyletic distribution. So you could search for groups that include and leave out specific taxonomic clades. You could also search for groups that have a particular number of proteins that might be most interesting in combination in a search strategy. So you might say as text term, and I only want to see groups that have, at least 100 proteins in them, or that encompass at least 100 different taxa. The orthogroup pages contain some summary information at the top, the total number of proteins in the group, the core and peripheral proteins, some keywords that occur frequently in the descriptions of the genes. If it's an enzyme, it may have an enzyme commission number. Um, Often there are PFAM domains in the proteins in the group, and then we've listed out previous groups from previous versions of OrthoMCL. Then we have this phyletic distribution that tells you what taxonomic groups this set of proteins in the ortho group are present. By default, we leave out the groups that don't have any proteins in this ortho group. And then we also have this table of all the proteins in the group. And this brand new feature that's only in OrthoMCL7, we've built a phylogenetic tree just of these proteins. I'm going to demo this in a minute to show you how to use the new phyllo tree. We've also added a feature called similar groups, new feature in Ortho 7, not in Ortho 6. Just in general, any sort of clustering algorithm has to make some tough choices. There's places where groups have to be split because they're just not quite similar enough. And there's cases where two subgroups are merged into one larger group, even though there's some distinct subclustering. But the overall similarity between the two clusters is high, and it doesn't meet the threshold for splitting. So large protein families, superfamilies, will typically be split by the OrthoMCL algorithm into a few or many groups. And you might want the superset of all the groups that are related to that protein family. So the similar groups feature works there. Also, sometimes you land on an ortho group, particularly if you're starting in a gene and you say, what ortho group is this gene in? And none of the proteins in that particular group have really interesting annotation. There's a lot of hypothetical proteins there. But you might look at similar groups and discover that there's a similar group that has a lot of well-annotated proteins. And so you could infer some annotation across similar groups. We also show a group statistic that is a measure of dispersion. How similar to each other are all the proteins in a group? Are there just a few outliers, or is the group very broad? I mentioned this phyletic search. We're going to go through this in an exercise coming up in just a couple of minutes. But there's this graphical tool that lets you select both large taxonomic clades or all the way down to individual species. And you can choose to leave in or leave out any set of organisms and say, give me the orthogroups that meet this particular set of taxonomic criteria. So, in this case, maybe a certain set of apicomplexans and leave out all the fungi, archaea, and bacteria. So I want proteins that, are not found in these organisms, but are found in these. Show me those orthogroups. As we saw earlier, every time you do a search, you get a result page. In ortho, typically you're searching for orthogroups rather than for genes. And a somewhat unique feature of orthogroups is that they have a taxonomic distribution. And in the results page where we're listing orthogroups here in a table, each group has its taxonomic distribution shown graphically. The color of the bars is just sort of arbitrary to help you visualize it, but how full the bar is represents what percentage of the organisms in that clade have proteins in this group. So if the protein is common or ubiquitous in all of the members of that clade, then you'll get a full bar, like for the euglenas here. But no archaea, no bacteria, and only a few alveolata have proteins in this group. This other group has a very different distribution where the bacteria have lots of bacteria have this protein. Um, what if you are working in a species that's not in ViewPathDB and therefore not represented in OrthoMCL? We've created a tool. Actually, it's been around for a while, but we've recreated it in a new way to map your list of proteins to the OrthoMCL groups. You just upload a FASTA file. We're imagining this would be an entire proteome from a newly sequenced organism, or it could be all the proteins that were generated in a metagenomics experiment, or some other list of proteins that's interesting to you and you want to know what OrthoMCL groups they fall into. You upload this FASTA file, you click this button, upload your FASTA file here, hit map proteins. We've not made this the search parameters configurable. We want to give you our best estimate of what group these proteins fall into. And it is more reproducible this way so that every time you would map this set of proteins, you would get the same result. And if you map different proteins, different FASTA files, you would get a result that uses the same search parameters. This is very sensitive. We're trying to give the match, as deeply as we can to things that are somewhat similar. So we use a pretty low blast stringency. We're using Diamond to run the blast search, which is a much faster implementation of blast, which still has a sensitivity down to about an e-value of 0.01. The results for your entire FASTA file can be downloaded as a tab delimited table with your query protein and then the best matching protein in OrthoMCL, the ortho group it belongs to, the keywords for that group, and the e-value. All right, I'm going to jump over to a live demo in a second just to show you how to use the taxonomic tree. So I'm here in the beta version of OrthoMCL where the tax trees live. And I'm going to start. I happen to know this sort of interesting gene family. It's a regulator and it's been well studied in Drosophila. It's called tailless. And I can just do a site search in Ortho for tailless. There's a bunch of protein sequences that presumably have known homology to tailless, but if I look at the ortholog groups, there's only two. And one of them is the nuclear receptors that I was looking for. And this other one, I don't know why it matches. Tailless complex polypeptide. So apparently tailless forms a complex with this other protein. I didn't know that, but we're going to look at this ortho group. It's got 900 proteins in it, which is kind of at the limit for what our tree allows. Has 261 core proteins from those 150 species that we know. The phyletic distribution, they're all metazoans. There's a lot in arthropods, 500 nematodes, other metazoa. Something that's interesting is if you open it up and you look at the individual species, we have a count for how many proteins are in that species. And this particular group varies a lot. This organism, Anopheles, seems to have only one copy. This 80s has 17. Very inconsistent across different species. I think that's interesting. Maybe that will be reflected in the tree. Um, Similar groups is interesting in this case also. We're looking at a nuclear receptor subfamily, and what we can see from the similar groups is that there are other nuclear receptor subfamilies. So in a sense, this is a superfamily of nuclear receptors, and the ortho groups are similar to each other. So when we're thinking about this group, it's possible that relevant proteins have actually been split off into some of these other similar groups. This is a tree that contains 900 proteins, and it's a little bit difficult to get a good mental picture of the entire tree just by scrolling up and down. So we've built these filters. One that I often use is just to limit it to the core genes. With 900 genes in the tree, it takes a moment or so to redraw. And then we throw up this little warning that this is a prune tree. So now we're not looking at the whole tree, but just at a subset of it. And this works somewhat. It's still a little bit too big to get a solid mental picture of what's going on with this protein family. How are the orthologs and paralogs related? So another way to do this, I'm going to take the core off, clear. It's going to redraw with all 900 rows. And then I'm going to look at Drosophila only. Come on. So Drosophila has 8 proteins in this ortho group, and they seem to be divided into 2 subgroups pretty nicely. This upper one is the 7-up, 2 different isoforms, E and F. And if you hover, you can get the full list, isoform A and C. So the phyllo tree represents it pretty nicely that this is the steroid receptor 7-up, 4 different isoforms. And this is the protein tailless that I started with, dissatisfaction, and another nuclear hormone receptor. How does that relate to the human nuclear hormone receptors? So we can add human to the tree, and it falls out pretty nicely that the tailless and dissatisfaction protein and hormone receptor 83 from Drosophila are all orthologous to this nuclear receptor subfamily 2, group E, member 1. So this human gene is orthologous to all 3 of these paralogs in Drosophila. So at some point in evolution, the arthropod lineage has made duplicates of this compared to human. And then over here, there's 2 other human genes that branch out. So these are paralogs in humans that are somewhat less closely related to these Drosophila genes. The 7-up genes over here are closely related to the human steroid 7-up receptor. So that seems to work out just well. Um, what about some other arthropods? Can we figure out when uh, when this gene's duplicated? So could we add, say, an 80s? Nope, no 80s. What else is in here that might be interesting? Yes, we'll add an 80s. And how do those genes match up with the Drosophila genes? Well, there it is. An unknown gene is, in fact, the 80s ortholog, probably of tailless. At least it's in that same branch of the tree. And that's kind of what I wanted to show, that you could use orthology and the phylogenetic tree to assign functions to unannotated genes. I'm gonna stop my demo and talk and let folks work on this themselves. So I'm gonna stop the share, ask for any questions for a minute. Omar, hi, yes.
I have a question.
Okay,
So
speak up.
if I have a protein which is a gene that is uncharacterized and would like to find an orthology in Drosophila, how do I go about it? Because you started with a known protein, tailless.
Sure.
But
Are
if
you,
I have to start with an uncharacterized,
is your gene in ViewPath DB?
yes.
So you go to the gene page, scroll
Mm-hmm.
down to the orthology section and click on the ortho group.
Okay.
And then you open up the ortho group list of proteins and you see if you can gain any information about the annotation of related genes.
Right.
And if you're in ortho 7, you can look on the tree and look at the genes that are most closely related to your gene.
All right. Thank you.
And we'll do that in our exercise in a little bit.
Yep. So that was a perfect question. Thank you.
Stuart, there are a couple of questions in the chat.
Okay, let's see if I can deal with it.
Why don't we move into the breakout groups and we can answer these questions in the chat so people can read them when they come back.
Also, if I get a minute, well, I'm going to be leading a breakout group, so I won't be able to answer chat questions.
Yeah, we have a minute right now. The breakout starts in 1 minute. So,
all right.
Stuart,
Well,
there's one question about the ortho-MCL classification. Yeah.
Yeah. So this one about, is it possible to add our own protein data and make our own trees? We don't have that feature right now. The upload of FASTA is designed to assign proteins to ortho groups, but not to add additional proteins to an existing group and rebuild your tree. So I would say what you would need to do is download the set of proteins from the most relevant ortho group, combine that with your list of proteins in a FASTA file, align them with Clustal, and make a tree using Clustal or some other phylogenetic tool. David?
Okay.
Yeah, but I also wanted to respond to this. I mean, it's an interesting question, and one of the questions that was asked was about the ability to classify metatranscriptomic data sets, and that was one of many rationales for the new for the current OrthoMCL algorithm, which both takes advantage of advances in OrthoFinder, and in particular uses an iterative approach to define ortholog groups based on core taxa and then add other sequences to that group. This is very appealing for metatranscriptomic analyses, and so that's certainly a functionality that we would like to be able to provide. It's quite straightforward and quite fast to be able to take large metatranscriptomes and add them in using this iterative process, where generating an orthology analysis based on an all by all blast comparison of all genes in a gigantic metatranscriptomic sampling would be both computationally challenging and highly misleading, because these do not include complete genomes and does not include a comprehensive sampling of all data from that sample.
Yeah. If I'm rereading the question, we don't have the tool packaged up so that you could use it yourself in a pipeline over and over again. You would have to submit your metatranscriptome FASTA file to our service, but I can say that it runs very quickly. I've uploaded a file with 100,000 proteins and get it done under a minute.
Okay, thank you.